This is the Algal Sample Collection Techniques lecture for ENR4345, Methods in Aquatic Ecology course at The Ohio State University. In this lecture, I will define periphyton and phytoplankton. I will then go over several different pieces of sampling equipment and how to use each of them. We will be looking at two different measures of algal biomass in this class. The first is periphyton, which is defined as benthic algae or algae attached to macrophytes. The photos on the left show a rock covered in periphyton, as well as a square where a scientist removed a known area of periphyton for study. The second type of algae we will analyze is called phytoplankton. This is algae that is floating within the water column. In the photo on the right, you can see green phytoplankton floating in Lake Erie. In this video, Hunter is demonstrating how we will be collecting periphyton samples in class. He finds a submerged rock with periphyton attached. He places the 2 by 2 centimeter measuring device onto the rock. Notice how he is holding the measuring device so that it doesn't move around as he scrubs using a clean toothbrush. When he is confident that he has scrubbed all of the periphyton off, he grabs his sampling bottle, which already contains deionized water, and swishes the toothbrush around inside the bottle to release the periphyton into the sample water. The grab sample is a simple technique that is demonstrated by Ethan. First, he will grab a small amount of surface water and shake it around inside the bottle to rinse all sides of the bottle with sample. He dumps out the rinse and simply fills the bottle with surface water from the sampling site. This video is showing how to use an integrated tube sampler, which is a long PVC tube that is often used in lentic systems to sample the top portion of the water column. This technique will capture the phytoplankton at the surface of the water, as well as the algae that are located deeper within the photic zone of the lentic system. Alessia and Kelly first rinsed the bucket and tube sampler with water from the sampling site. You will notice that they do not touch the inside of the bucket. This is to prevent contaminating the sample with anything that might be on their hands, such as lotion or sunscreen. Next, the sample is collected by putting the integrated tube sampler into the water column to a marked length and then adding the plug to the top of the integrated tube sampler. Finally, they will pull up the tube and collect the sample within the bucket. The next sampler we're going to learn about is called a Van Dorn sampler, which is commonly used in lotic systems to collect a sample of water from a certain depth. Natalie is demonstrating how to set the Van Dorn sampler in this video. First, she opens the doors on each side and connects them with pins at the top of the sampler. Next, she would lower the Van Dorn sampler to the desired depth, which was not done in this video for demonstration purposes. The metal weight that Natalie is now holding over the sampler is called the messenger. The messenger is released down the string, and when it reaches the Van Dorn sampler, it pushes a button that snaps the watertight doors on either end of the sampler closed. Finally, the Van Dorn sampler can be reeled in, and the technician has a sample of water from a discrete depth within the water column. Van Dorn samplers can be horizontal or vertical and come in a variety of sizes. On the right, we have a rosette or a carousel sampler coming aboard the RV Tioga. This computerized sampler has several vertical Van Dorn samplers attached and will often have other apparatus for measuring water quality parameters such as temperature or dissolved oxygen. The operator will lower the sampler to the intended depth while reading the water quality parameters, and then based on that data, will tell the computer at what depth to close each of the Van Dorn bottles on the way back up to the vessel. Deckhands will help the operator bring the array back onto the vessel, stabilize it, and then collect water samples from the Van Dorns. Finally, we have the plankton net, which is used to collect phytoplanktons in systems where the water is deep enough to submerge the net. Plankton nets can be used either horizontally or vertically, and there are several different sizes of nets available on the market. At the bottom of each plankton net is a collection cup with a mesh filter on it. The mesh on these cups come in different sizes. A smaller size mesh will be ideal for phytoplankton collection, but a larger mesh will allow smaller organisms to pass through the net, so we can collect only larger organisms such as zooplankton. This is called a flow meter. It has a propeller on it and a set of numbers on the side. When deployed, it hangs down from the middle of the net and spins as the net is drawn upward while collecting a sample. You can use a flow meter to tell how much water actually went through the plankton net to calculate the volume of water sampled with the net.